I've only ever worked on rare bone disease. That's been my entire research career. So I'm thinking back to when I was 21 trying to clone the collagen 1 alleles from a young kid with osteogenesis imperfecta. Fast forward in 30 years, we're now beginning to start a clinical trial on another collagenopathy. And I just hope it doesn't take the next 30 years for the next trial. So I work at Newcastle. So for anyone who's not been up to Newcastle, we've got a very good setup there. I'm based in the Centre for Life. If you've ever caught the train into Newcastle, after you've seen the fantastic bridges, it's the first building on the left. It's this big yellow and blue coloured building. And really, it's a combination of the Institute of Genetic Medicine, which is Newcastle University, and the Northern Genetics Service. And it's that combination of clinical and basic science that really allows us to progress our research findings from bench to bedside. And in fact, we're quite a strong hub in many different rare diseases in Newcastle. So my interest for the last 30 years really has been on this group of rare bone conditions which we know as the skeletal dysplasias. There's extensive clinical and genetic heterogeneity, and we think there's about 450 different forms of chondrodysplasia. Although they're individually rare, as a combined group, they probably have an incidence of about 1 in 4,000. Put that into context, cystic fibrosis is probably 1 in 3,500. So really, as a group of diseases, they're reasonably common, but some of them can be ultra rare. The one that I'm going to talk about is about 1 in 100,000. Some can be a little bit more common, such as achondroplasia. Even to date, genetic skeletal diseases are difficult to diagnose, particularly for the non-expert, and there are currently no treatments or biomarkers available, and therefore, obviously, this confers a great burden, both in health, pain and disability, and high healthcare costs. So what I want to talk about, really, and I'm not going to bore you with the last 30 years, I'm just going to talk about the last 2 years, really, which started from work that was completed about 2 years ago by a colleague of mine in Manchester. When I used to work in Manchester, we've been working together for many years, looking at rare bone diseases in mouse models. And Ray, and I'll talk more about this, completed the clinical study, preclinical study in September 2015. It so happened that at the time, he sent me the preprint to read, well, the first draft to read through, just as our tech transfer officer was in my office, and I showed it to her, and she said, that looks really exciting. And immediately, the university moved to get a UK priority patent. So this addresses your question of IP. The university, Newcastle University, is very keen on rare disease research and a translation of rare disease research. So we filed a patent covering the use of this drug carbamazepine in a broad group of rare diseases in which we think they share a common disease mechanism. So I think that's probably the strength of the patent. We obtained Orphan Drug Designation for carbamazepine about a year after we'd completed the preclinical study. This was important, as become clear. A year later, once we've done a few more studies and showed that carbamazepine actually did work for a few different diseases, we filed an international patent. It took about 2 years for us to publish this preclinical data, not because we were slow. We aimed for the high journals and they kicked us down. So we finally ended up in Journal of Clinical Investigation, which is still a very, very good journal. And where we showed our initial preclinical work was in 1 mouse model, we ended up having to do it into 2 mouse models to get it into JCI. So after getting the Orphan Drug Designation, having this preclinical data allowed us very fortuitously to apply for Horizon 2020 funding in the recent call, and we were awarded that, and the contract was signed in December last year. And at the same time, it was also a requirement to get protocol assistance from the European Medicines Agency, which we did again in September. So everything's now set up to begin this clinical trial. So what I want to do really is run through some of the stages that we've been through over the last 2 years, starting, of course, with showing some of the preclinical data. I'm a mouse geneticist, so I've got to show pictures of mice in a bit. So as Rick's already mentioned, metaphyseal chondrodysplasia type Schmid, it results primarily from a disruption to endochondral ossification, particularly affecting the metaphyses, and you can see this flaring and irregularity of the metaphyses. The disease is also characterized by this acoctivara, this deformity of the hips. MCDS results exclusively from mutations in type 10 collagen, and type 10 collagen is expressed exclusively by hypertrophic chondrocytes in the growth plate. So this, here we've got the epiphyses and the metaphyses, and here we go with the epiphyses, the metaphyses. Then you see that collagen 10 is expressed in this one cell type in this very precise region. And therefore, this is why we see a metaphyseal dysplasia, because of localization of collagen 10. And all patients with MCDS have a collagen 10 mutation. So about 15, 20 years ago, we made the decision in Manchester to really work on mouse models. If you've ever worked on the growth plate, or if you've ever worked in skeletal diseases, really the only way to do this is through the mouse model. It's almost impossible to model in a cell-based system. So we started generating relevant mouse models about 20 years ago. We generated one for metaphyseal chondrodysplasia type Schmid. We knocked in a point mutation we'd identified in the family. And the mouse model very nicely recapitulated the phenotype. It was obviously short. And also we saw a hip dysplasia, hip abnormality that was not too dissimilar from the patients. Based on having had this mouse model for the first time ever, we were able to really begin to dissect the disease mechanisms. This staining, so staining in black, as I've mentioned, is collagen 10. So collagen 10 is an extracellular matrix protein. The cell makes it and it secretes it, and it performs its function outside of the cell. What you might see here is a difference. So this is the wild type, and this is the mutant. And you can see that there's a lot less staining, but what staining there is actually within inside the cell. So the collagen 10, because it's got a mutation, misfolds, it's retained within the cell, and that has profound consequences for the cell. So what we see is that, so this is a marker bit, this is a marker for something called ER stress. Because the protein is retaining, the cell begins to swell, and that retention of that protein, it's effectively constipated, and it stops that cell performing its normal function. And in this case, its normal function is to direct the growth of the bones and cell differentiation. So what we see is a widening of the growth plate here. So this is a classical pathological feature of Schmid, the widening of this growth plate, due to the retention of mutant 10 that causes ER stress. So of course, ER stress now gives us a mechanism that we can target therapeutically. And it's a very attractive mechanism. We know it's a core disease mechanism, MCDS, but also in a broad range of other bone diseases, but also other connective tissue diseases, and a broad range of diseases, cardiovascular, neuromuscular, macular degeneration. So it's a common mechanism that can be pharmacologically targeted through the use of drugs and small molecules. And we'll be looking to maybe decrease the misfolding of the collagen 10, or increase the folding capacity of the cell so it can actually deal with this mutant protein. Or perhaps the best one is to help the cell degrade the mutant protein. So if you can degrade the cell, if the cell can degrade the mutant protein, the ER stress goes away and the problems go away. So this is one of the first experiments. This was performed by a student that Ray and myself supervised in Manchester. And all that Lorna did was to take a cell model, so this is a cell expressing mutant collagen 10, and treat with a range of different drugs. Sodium phenylbutyrate, carbamazepine, a bile acid, a flavonoid from red wine. So these are just drugs that we'd read in the literature are able to alleviate ER stress. And what we saw in our cell model, here we've got 3 markers of ER stress, that carbamazepine and carbamazepine alone reduce these 3 markers of ER stress. So we knew that the drug carbamazepine was targeting ER stress in this cell model. We were able to quickly go on to treating the mouse model. And here we have, in the wild type, this is MCDS with this characteristic widening of the growth plate due to changes in cell phenotype. As soon as you add the drug, you can see that the width of the growth plate immediately almost resorts to normal. This bit, again, a marker of ER stress. See no ER stress in wild type, lots of ER stress in the cells in the mutant growth plate, which is again reduced by carbamazepine. And again, we see that the carbamazepine is acting by reducing the amount of retained collagen 10 in cells within the growth plate. And what I have not shown, but what I can tell you is that we know that carbamazepine is inducing the degradation of mutant collagen 10. And of course, if we're working on a rare bone disease, we want to see that treatment improves growth velocity and bone length. And in fact, it worked remarkably well in these mouse models. Within 3 weeks of treatment, bone growth was almost restored to normal. So why were we starting to get interested? Well, carbamazepine is a drug, and it's been known for a long time. First discovered in the 1950s. It's been used in epilepsy since the 1960s. It's a generic medicine. It's not very expensive, and is in fact within the World Health Organization's lists of essential medicines. We think that it costs about 5 pence a dose. So this is a drug that's as cheap as chips that works on a very, very rare bone disease. So On the basis of the preclinical work, we approached the European Medicines Agency to get Orphan Drug Designation granted for the use of Carbamazepine in MCDS. It's quite straightforward, really. So we've got excellent preclinical data. It's a rare disease, 1 in 100,000, and of course there is no treatment currently available. And we tick all boxes for Orphan Drug Designation. It was quite a quick process. I guess administratively wise, it was a big form, so we had to employ an expert to help fill out these forms, which cost a few £1,000s. But other than that, it was a very smooth process, really. So having preclinical data, having Orphan Drug Designation, we then began to embark on trying to get funding for this study. It so happened at the time that a call came out, the European Commission Horizon 2020 for personalised medicine. And this call really offered an awful lot of funding for rare disease research. We were particularly interested in this one, new therapies for rare diseases, in which €65 million was available. The only problem is these are quite competitive. So the number of proposals, it's a two-stage process. The number of first-stage applications received for all 4 calls were 668, of which only 100 reached the threshold to go through a second stage. So it's a very heavy cut-off at the first stage, but this only requires about 6 pages pre-application. Because once you get to the second stage, there was 97, and about just under 40% were funded. So if you're through to the second stage, the odds improve dramatically. I'm led to believe that for this particular call, 127 applications were initially received and they funded 10 projects in the end. So it is quite tough, but once you get to this stage, certainly got a better chance. Part of the process of being able to apply for funding was the requirement to have sought protocol assistance from the European Medicines Agency. Now, a problem arose with that in that no one in Newcastle knew how to do that. So we took the wording, the grant instructions, that we had to have approached them. So we approached them, submitted the grant, and then withdrew our application. Then employed an expert to help us write this. And it was the same expert that had got us the Orphan Drug Designation. Cost about £90 an hour and worth every penny. So we put our letter of intent in September. It went to and backwards. We submitted our questions that we wanted the approval on. They sent back a written reply. We addressed it. Had to go down to the European Medicines Agency. We had a 2-hour discussion, which was quite amusing if you're a biochemist and they're expecting a clinician there. So, but nevertheless, we went through the process. Our questions were really based around 3 points. Our statistical size, and we talked about statistics earlier this morning, and the sample size. Say it's a rare disease, we're only ever going to probably recruit 40 patients. The big discussion about, was it going to be a double-blind placebo, as Tracy mentioned earlier this morning. We went for an open label. And the EMA initially didn't like that, but when we pointed out that this is a really rare disease, they actually agreed to us having an open label clinical trial. The proposed primary endpoint again caused a bit of a problem with the EMA. We wanted it to be pain, because pain is really the major symptom with these kids. They thought it was too subjective and suggested that we had growth velocity, so we agreed. We changed it to growth velocity, but then they suggested, well, actually, if you've got abnormalities, if you're going to make the bones grow faster, you're just going to increase that abnormality. So we all agreed that actually the endpoint has to be a combination of everything, really. We've got to look at pain, growth velocity, and joint misalignment. So I'm happy to say, after that meeting, I didn't mess up too much, and we got protocol assistance. The EMA charged for protocol assistance €63,000. We got a 75% reduction because it was an orphan product, and then they waived it because it was a paediatric advice. This is pretty important because coming up with €60,000 is not easy, particularly before we'd got the grant awarded, so we couldn't charge it to the grant. Nevertheless, so we ended up having that for free, but the expert advice would probably come out at about £8,000. But without Steve, we just didn't have the expertise in Newcastle University to do this. So it's certainly a consideration. So moving on to the trial itself, Rick has already mentioned it. There's really 4 phases to it. We think it's a phase 2/2B clinical trial. We've got a big health economics component because I think it's important that we really know what is the cost of rare diseases, rare bone diseases, and will a drug make a difference. Now, if that drug's only going to cost 5p, it's a no-brainer, really. Obviously, as Rick has mentioned, this public patient engagement is incredibly important. And really, I think one of the most important things is biomarker discovery. I think that this is becoming incredibly clear how it's really important to have biomarkers, particularly when you're discussing endpoints and you're looking at growth velocity. If you've got a biomarker, it's going to make life a lot easier. So this is our plan for the clinical trial. As I said, it's open label. There's one other thing I should mention. And I'll be interested to see how other people have addressed this. Sponsorship of a clinical trial. When it's multi-centre, multinational. The NHS can only sponsor clinical trials in the UK. So we've not got a backing of a pharmaceutical company who would naturally sponsor the trial. So we've had a little bit of problems with sponsoring the trial. So the NHS and Newcastle hospitals will sponsor the UK part of the trial. So what we're going to do for the first stage is we're just going to recruit patients in the UK. So that's ourselves in Newcastle and Melita here in London. Because it's open label, we're going to do a 12-month baseline analysis, looking at growth velocity, pain, quality of life, and so forth. And at this time, also collecting serum for biomarker analysis. We're then going to do a dose finding, 12 months of dose finding. So this is just on 12 patients recruited in the UK. Now, the dose finding, we're thinking should be quite straightforward. We know what the maximum level of carbamazepine you can give to kids. We can correlate what effect we saw in vivo on our mouse models. But probably the best is that maybe about 15 years ago, 2 groups in South America treated 3 kids with a bone dysplasia called Schwartz-Jampel disease with carbamazepine. They were treating them for myotonia, but noticed an increase in height. So we've got the dosage that those 3 kids were given. So we're thinking that we can very quickly go to, I think it's 20 mg per kilogram per day. So that's it. We've done the dose finding. So we've got this 12 months of dose finding. At the time that we're doing the dose finding, the other patients will be recruited in these other centres here. So Antwerp, Paris, Freiburg, Bologna and Melbourne. At which point, we are hoping to have resolved the sponsorship problem. And it's looking, hopefully, that Newcastle University will sponsor the entire trial, at least this stage here. And then these 2, so the baseline from these patients, and then the 2 will converge, and we will treat all 40 patients with the dosage for 2 years, measuring the growth velocity, joint misalignment by X-ray, pain, and possibly by then, we'll be 24 months in, we may have a biomarker. So that's one little story on one disease, and I said there's about 450 different chondrodysplasias, and we've been fortunate over the years, really, as a rare bone disease research community, to have had substantial EU funding through all of the different frameworks. Initially, it was through a network to start to diagnose and identify the molecular and genetic basis of these diseases. More recently, we had 2, an FP6 and FP7 program. The FP7 is still running, where we really started to develop our mouse and cell models and begin to study them in depth. So currently, through this project, we generated about 100 different cell models and about 35 different mouse models, looking at disease mechanisms, trying to group dissimilar phenotypes into similar mechanistic groups and starting to do the preclinical drug screening. So I've spoken about MCDS. I'd say there's upwards of 50 or 60 conditions that we're looking at with a view to starting to screen using FDA-approved libraries from, for example, the Presswick library or the one from MRC Technology. And really, this most recent grant has taken some of the work that we've done on FP7, FP6 through to the clinical trials. So we've really seen from the bench to the bedside with this particular condition. I said there are others in the pipeline. So really, just to finish, this is our new logo, which we're going to have to adapt slightly. I'd first like to thank, so really the brains behind all of this, all of the science, was my long-time friend and colleague, Ray Boot Hamford, who's been working on collagen 10. They cloned the first gene. Not only did they clone the gene, they isolated the protein collagen 10 from chick back in the 1980s. So it's Ray really that came up with the idea to start to screen for using carbamazepine. It was Lorna that the PhD student said that actually did the work. So it was very much Ray's idea. He's now retired and handed over the clinical trial to us really in Newcastle. So Michael and Marta, who are clinical geneticists, who again I've known for a long time, been very instrumental in the approaches to EMA and starting to pull together a protocol that Melita will now be helping us with and will develop through the clinical trials unit. Laura and Joe, who again pretty much pushed the IP part of this project and making sure that it's protected. And of course, I would like to acknowledge the funding we've just received from Horizon 2020, which is, I think, just over $5.5 million for the next 5 years to deliver Carbamazepine as the first non-surgical treatment for MCDS. So thank you for listening.